This is the Human Geographic Diversity lecture. In the last lecture, we discussed the genetic component of diversity, where it comes from and how it is introduced into a population. Now that we have a better understanding of genetics and the ways in which variation can be introduced into the genes, we need to move on to discuss how we understand population history from genetics. In this lecture, we are going to discuss variation within and between populations. If you remember in the first lecture, we discussed Lewontin's 1972 research, where he showed that more genetic variation exists within a population than between two populations. In this lecture, we're going to actually discuss how this is measured. We will also discuss what evolutionary forces can affect population variation and how each force can either increase or decrease variation in a population or both. When we begin assessing variation within and between populations, we are doing what's called population genetics. This is an entire field of study. And I've defined it here on this slide. It's the quantitative study of how genes are distributed within and across populations. But before we really get into some other definitions, I think it would be useful to give you an example of what population genetic studies do and the sort of questions that these type of studies are essentially trying to answer. Something I discuss in my introductory course is sickle cell anemia, and this is something that we're going to kind of keep coming back to throughout the semester for various examples. A lot of people assume that this is a disease that is racial. However, this is another topic altogether, and we're going to discuss this later in the semester. But sickle cell anemia is an example that I typically give in my introductory course for what is called heterozygote advantage. This is a genetic phenomenon where one genetic type, in this case the heterozygous form, is being favored in a population. And I think I briefly mentioned this in the last lecture. But sickle cell anemia is a deficiency of the red blood cells. So typically in a normal individual that is not affected by this disease, their red blood cells are going to be normal cells. So they're going to have a really nice round shape to them. And you can actually see a drawing of what we would consider to be normal red blood cells on the PowerPoint slide. This is important because red blood cells are the components, the things that carry oxygen throughout your body. So having them be nice, plump, round, healthy cells will allow them to be able to do this efficiently, to be able to carry oxygen throughout your body. If a person is affected by sickle cell anemia, their red blood cells have a sickled shape. So you can see in the PowerPoint slide a picture here of a slide of red blood cells where most of them are sickled and only a few of them are actually have a normal shape to them. For these individuals that are affected, it's often deadly because obviously their body's having issues getting oxygen, but their body is not able to function properly because they're not effectively carrying oxygen throughout the body. So if a person is normal for the trait, we could classify them as a homozygous dominant individual. So let's just say we'll use A's for this particular example. A homozygous dominant, capital A, capital A. If they have sickle cell anemia, then they are homozygous recessive for the trait. So this would be a lowercase a and a lowercase a. If they are heterozygous for the trait, they would be considered what we call a carrier of sickle cell anemia. Carriers, therefore, have some cells that are normal and then they have some cells that are sickled, so they kind of have a little bit of both. But what's really interesting about this disease is that research has shown that there's a geographical pattern for its occurrence. The presence of heterozygous individuals is very prevalent in areas throughout Africa. There's a reason for this, and we're going to discuss it in more detail later in the semester, but let's relate this example to population genetics. That's really the only reason I'm bringing this example up right now. First of all, this type of knowledge, knowing that this particular form of genetic loci occurs more heavily in a certain geographical area, is due to population genetic research. But you can also use this type of example to understand the research that is conducted. For instance, okay, so African populations carry a higher frequency of this allele. So some questions you would want to ask: is this due to chance? Is this due to cultural practices? Not only that, but what has played a role in creating an increased resistance to this allele in other populations? Why is it that the heterozygous form is being favored over the homozygous dominant form, since obviously it would be more advantageous to have all normal cells? So these are the types of questions that population genetics is designed to answer. The entire purpose of population genetics is to better understand the variation of alleles within a population, but to also understand why and how have these alleles changed over time, and how have they responded to forces, evolutionary forces. So we're going to look briefly at how population genetic studies are actually conducted. First, I want to start with just some simple definitions. I keep using the term population, so we need to actually define what I mean by population. A population consists of reproductive age individuals that may contribute offspring to the next generation. Defining which individuals belong to a population can be quite tricky, but if a population is isolated, then it's very clear, obviously, who their members are. However, once you begin distributing populations continuously across the landscape, then your boundaries of one population versus another can get kind of blurred. When assessing or measuring how much genetic variation exists within a population, or when comparing the variation between two populations, geneticists use probability theory. Probability theory is a way of measuring the frequency with which events occur, or stating the likelihood that they will occur, and relating that the probability of specific events. So when assessing probability, there are two rules that are used. These rules allow us to see how individual probabilities can be combined. So let's start with understanding the OR rule. This rule is used when you are trying to assess the probability that one or the other will occur. One event or the other will occur. For example, it would be used to ask the question, what is the probability that a GH heterozygote will produce a G gamete or an H gamete? Keep in mind that this rule does not allow for the two events to happen together. The AND rule is used to find the probability that two independent events will occur. So an example would be to assess the probability that a GH heterozygote will produce two gametes, a G and then another G. So when geneticists use these rules of probability, they assess population variation through the Hardy-Weinberg Equilibrium. This is a mathematical construct that allows geneticists to predict the structure of a population over time if certain criteria are met. In other words, it provides a baseline expectation for a population to not change, to not undergo evolution. So I've gone back and forth about how detailed I want to get in terms of making you all calculate this equation, and I've decided that for this class I expect that most of you have already done Hardy-Weinberg calculations in the past, so I'm not going to get into the calculations, but I am more concerned about you knowing how the Hardy-Weinberg is used as a mathematical equation. How is it used to assess variation in a population? So as I've said, the Hardy-Weinberg equilibrium is just a baseline expectation of what we would expect the genotype frequencies to be if a population was in equilibrium. So to do this, using your known allele frequencies for the population, you would then calculate the frequencies that you would, quote, expect using Hardy-Weinberg. So again, if you haven't done this in a previous class, this is basically a lot of mathematical calculations that you would use to determine these numbers. So the expected frequencies are the numbers that you would expect if the population is not undergoing evolution. So let me say that again. The expected Hardy-Weinberg frequencies are the allele frequency calculations that you would expect if the population is not undergoing evolution. So on this slide, I've put an example of some frequencies for a population when the three genotypes for a trait are assessed. MM, MN, and NN. As you can see, the expected frequency for the MM genotype is 0.25, and the actual frequency for the genotype in the population is 0.24. So expected frequencies are what you would expect the population to, the frequencies that you would expect the population to have if they are in equilibrium, if they are not undergoing evolution. The observed or actual frequencies is what you are actually observing in the population. So as you can see, 0.25, 0.24, these two frequencies aren't really differing that much from each other. You can also compare the other two genotypes and see that the expected and the observed frequencies do not differ greatly from each other. So what this means is that for this particular trait that is being observed, the population is not undergoing evolution. That's because the actual frequencies that you're observing are fairly close to what you would expect the population to be if they are not undergoing evolution. So if these numbers were drastically different from one another, let's say that expected frequency was 0.75 and our observed frequency was 0.25, then this would indicate that this particular genotype is not in equilibrium and that something is affecting that genotype. Some type of evolutionary force is causing that genotype to undergo evolution. So what does this all mean? Why is the Hardy-Weinberg important? Why is it used to assess variation? Essentially, when we say that a population is in equilibrium, we are saying that some trait that describes that population is remaining unchanged through time. If the population is deviating from Hardy-Weinberg, this is what's interesting. This is what tells us, because these are the traits that suggest that some type of evolutionary forces are at work in the population. These forces are those that we have already discussed and include mutation, selection, drift, and migration. I want to briefly discuss how each of these can change allele frequencies. And again, this is a really simplified version of population genetics and quantitative genetics, but I just kind of want to give you the basics of assessing a variation in a population so that we can move on to really getting into the details and the more, I guess, to me, interesting discussion of human variation. So mutation, as we previously discussed, this is the only way that you can introduce new alleles into the population. The rate of this can be measured using Hardy-Weinberg calculations, but for the most part, the effect of a mutation on allele frequencies is really small. So when compared to the other forces, mutation is considered to be a weak force in that it doesn't change allele frequencies at a high rate. In other words, it really isn't a force that will pull a population that far from Hardy-Weinberg equilibrium. The only way it will have noticeable effects is if it occurs over a long period of time. Mutation is also one that acts in conjunction with other forces, meaning that the other forces of evolution operate with mutation to produce changes in allele frequencies. Most mutations are considered to be deleterious, but there have been advantageous ones that occur as well. Selection is a force that must operate on genetic variation. So as I've previously mentioned, mutation is the precursor for evolution to occur through natural selection. Also keep in mind that selection is occurring within a population because there's some type of differential survivorship. So if we are looking at a particular trait in a population, let's just use stature as an example, then how the population responds to selection is determined by where the population is most fit or where it shows the greatest survivorship. So there are two categories of selection that population geneticists consider: directional selection and balancing selection. Directional selection occurs when one allele is being favored. So let's go back to our example using height as a trait. This would be as though one extreme is being favored, maybe tall individuals. Balancing selection occurs when two alleles are being favored in the population. So this would be as though the average height, not short individuals, not really tall individuals, but just your average height is being favored. The most common cause of this is heterozygote advantage. And when this occurs, an individual that has two different copies of an allele is more fit than individuals with one copy of that allele. For example, let's say we are referring to allele G. Individual with heterozygous for this trait, capital G, lowercase g, will have a higher fitness than individuals with both the homozygous form, whether that be dominant or recessive. The heterozygote individuals are favored over both homozygous forms of the trait. Because the heterozygous individual is being favored, then both alleles are going to be maintained in the population. Two rules to remember about evolution through selection. Evolution by natural selection can occur rapidly if the dominant allele is being selected against, while on the other hand it will occur slowly if the selection is against the recessive allele. So why is this? Well, it helps me to remember this rule by thinking about the phenotypes. Remember, when viewing the phenotype, the recessive alleles are essentially hidden in the heterozygote. So, for example, let's just say we are talking about selection against plant height. Maybe we have a field full of plants, and some of them are really tall, some of them are really short plants. Let's argue that, or let's denote a capital T for tall plants and a lowercase t for short plants. If there was a hungry goat that was only eating every tall plant in the population, in other words, selection, the goat, is acting upon the tall plants, then in one generation, all tall plants could possibly disappear. Selection, which is the goat here, remember, is only seeing the phenotype. The goat is only seeing tall plants, and therefore evolution in this population would be really fast. If selection was against the short plants, this would slow evolution down because the goat would eat all of the plants that are homozygous recessive. But the recessive allele wouldn't actually disappear from that population because it still exists in the heterozygous dominant plants, which are the tall ones. In fact, this allele can't be completely eliminated because of this. So keep in mind that recessive allele is being hidden in the heterozygous form of the trait. But all you're seeing, the phenotype, is what's being seen by the selective pressure. This moves us to discussing the effect of genetic drift. Remember, genetic drift is a change in allele frequencies within small populations that occurs across time. This is due to chance events, so it's a completely random force. It can have significant impacts on the evolution of a population if the population is small. If a population is large, you're not going to see this random genetic change in terms of evolution. You just won't see it. Why is that? Well, a good way to think about this is to imagine that you have a population that's represented by three colors. So you can see here in the first picture on the left side of the slide, you have red, yellow, and blue beads in this jar. Let's just imagine that these colors represent all of the possible alleles in the population. So lots of different variation, even though there's only three colors we're kind of looking at here. Let's just pull out a small handful of those beads, and then you can move to the second jar, the second generation. So now the frequency of these alleles are not equal to the original frequency that we had. You can see there's less reds, a lot more blues. Another handful taken out of that jar, and you continue to minimize the frequency of the alleles in each generation. So through time, this is randomly shifting. There's no pattern, there's no process to this, but it's randomly shifting the frequency of alleles and will produce drastic evolutionary impacts on the population. So for example, you can see here in the last picture of the jars that the population now only contains blue and yellow beads, and it's skewed toward a larger number of blue ones. This is a completely random process, but it can have a really drastic impact on the population. It is common that when genetic drift occurs in a population, there tends to be a deficiency of heterozygotes and an excess of homozygotes. This leads to an interesting interplay with other forces of evolution, like mutation, since typically mutation will act to increase heterozygosity. Lastly, migration is a force of evolution that affects population variation. When the term migration is used, though, it's essentially referring to gene flow. So Keep in mind that gene flow is considered to be one of the 4 forces of evolution. But when we're talking about population genetics, we typically use the term migration for this. So this is a movement of alleles. There is a genetic phenomenon that occurs with migration referred to as isolation by distance. So when you think of 2 populations exchanging genes, typically one population will exchange genes with their nearest neighbors. So they're not going to go further than they have to. But the population that is so they're going to exchange genes with their nearest neighbor, the population that's living closest to them. So this is going to lead to populations that are closer together in terms of geography. They are going to be more genetically similar to one another than populations that are further apart. So this is what is referred to as isolation by distance. So migration actually acts to increase variation within a population, which makes sense if you think about its definition. Essentially, the allele frequency of already existing alleles has the potential to increase. Or you could introduce new alleles into the population if there's some type of new mutation being introduced from the neighboring population, from the migrating population. I want to finish this lecture with a discussion of the assigned article for the week. And something you are probably already realizing is that the articles that are assigned each week in some way relate to the lecture, but they are chosen also to move us forward to our discussion for the second portion of the course. I'm not sure if you've realized, but the second portion of the course is a little bit heavier in lectures and heavier in material, and a few more weeks longer than the first portion of the course. The assigned article for this week is by Jared Diamond, and if you haven't read it, please be sure to do so. But I wanted to briefly summarize this article and just kind of talk about a few points that relate to what we have discussed in this lecture. First of all, one of the main purposes of this article, and something you should be kind of thinking about as you're reading, is that Diamond's trying to look at answering the question of how is it that traits become geographically patterned. And Diamond focuses on looking at skin color kind of as the main example throughout the article. But the answer to this question can be applied to all sorts of different traits. He mentions some ways in the article that geographical patterns can occur. First, one is through natural selection. So this is a force that we already discussed, but variability is first produced in the population due to some selective force acting upon it. And he actually provides an example of the differences between northern hares and weasels and the southern ones. The northern species produces white fur during the winter time because selection is providing them with a camouflage throughout the winter months. But for the southern species, selection maintains this brown colored fur year-round. Traits can also vary geographically due to sexual selection. This is something that we didn't discuss in this lecture, and so I just want to briefly mention it here. But sexual selection is a lot like natural selection. It's essentially the same thing. It can do the same things in terms of heterozygote advantage and stuff. It can do those same things in a population. It's just how it comes about is different. It's like natural selection, except that traits are being selected due to individuals of one sex being attracted to mates of the opposite sex based upon a specific trait. So depending on what trait is being selected, this can create differences between populations. So take note of the examples that Diamond provides for humans, and he gives a number of them. examples for sexual selections and also sexual selection, and also examples for natural selection. So I just wanted to point out that these are really good things to focus on throughout the article, as they make for good test questions. So that wraps up this lecture, and I've tried to keep this lecture as simple as possible, but I do realize that most lectures on quantitative genetics is all math. And I just really want you to understand how genetics plays a role in producing population variation, and how is it that those various forces of evolution act to increase or do they decrease population variation.